We do have a student speaker this evening. It is Vaithish Velazhahan, who is a research fellow at Caius College and is going to give an online talk on developing novel antifungals, lesson from G protein coupled receptors. Vaithish, thank you very much. I can see you on the screen, and I shall now sit down and enjoy your talk.
Amazing. Thank you so much for the kind introduction. Let me just share my screen. Yeah, hello everyone. Thank you so much for the opportunity to share my PhD work today. I'm going to first introduce you to a class of proteins that I study called G protein coupled receptors, also nicknamed as GPCRs. This class of proteins represent one of the largest family of membrane proteins in humans, and they are present on the surface of the cells. And they're very important to allow cells to communicate with an organism's environment. They are called GPCRs because they perform their functions by activating a downstream protein called the G protein. And that's why they are called G protein coupled receptors. So you can see that they are one of the most successful drug targets, and about 35% of all FDA-approved drugs target just this family of proteins, and they're implicated in a wide variety of different diseases. GPCRs could be broadly classified into 6 different classes, of which the classes A, B, C, and F represent the mammalian and human GPCRs. And these squiggly lines right here, for those of you who don't know what they are structures of proteins. And we have hundreds of structures of human GPCRs that have been determined. And these structures greatly facilitate drug development, as I will explain you shortly. Whereas there were no structures available for the fungal specific class D GPCRs when I started my PhD. So the goal of my PhD was to try and determine the first structures of fungal GPCRs. The fungal GPCR that I worked on was called ST2, which is a prototypical fungal GPCR from a yeast called Saccharomyces cerevisiae, better known to you as the yeast that's responsible for making alcohol and bread. The goal was to determine the structure of ST2 from this yeast, and this particular GPCR is very important for sexual reproduction in fungus because it senses a sex pheromone that comes from the opposite mating type. Just as humans have a male and a female, fungi have an alpha strain and an A strain. In this particular case, for the two to come together and undergo sexual reproduction, this particular receptor was very important. For the past 2 decades, many different groups have tried to use different approaches to determine the structure of ST2, but because of various technical limitations and challenges, it remained impossible. And the goal of my PhD was to develop new techniques that would then allow the determination of the structure of ST2. You may wonder, why should we care about fungal diseases in general? Fungal diseases present one of the major threats as they kill roughly 1.6 million people annually, which is literally about the same as that of tuberculosis and about 3 times higher than that of malaria. They also destroy 25% of the world's food production, which is estimated to be enough to feed roughly 600 million people every year. Majority of the currently used antifungal agents belong to some of the limited classes, and in many cases when the disease becomes life-threatening, the existing agents are either ineffective or the fungi have become resistant to them, which means we need new classes of antifungal drugs. And this problem has been declared in 2022 as a global health emergency by the World Health Organization, WHO. We know that GPCRs are one of the best drug targets in humans, and consequently, it has long been proposed that GPCRs in fungus could also be targeted to make the next generation of antifungal drugs. But because of the lack of structures of these proteins, it was not possible to do that. And the goal of my work was to try to solve this problem and potentially use GPCRs to make new antifungal drugs. You can appreciate from this Venn diagram that in many common cases of human death, such as tuberculosis or cancer, asthma, or AIDS, in several cases, it's actually a fungus that infects the immune-suppressed patient and eventually kills the patient. Majority of these patients who have comorbidities have weakened immune systems, and the fungi takes advantage of it and infects and kills the patient. 2 examples from contemporary news includes the fungal outbreak that is vanishing the majority of the bananas that has remained in human consumption for the last 50 years or so. So the world's most favorite bananas comes from a single variety called the Cavendish bananas. And it turns out that over 99% of this particular strain of bananas have gone extinct thanks to a fungal disease which has been rampant in killing all the Cavendish bananas. And another example is the latest mucormycosis outbreak in India during COVID, which was very rapidly spreading, and there was no treatment for this particular disease, and it killed about 5,000 people in the end, and it also destroyed the lives of many, resulting in serious injuries to health as well as to property in over 50,000 people in total. So this was a very devastating disease which was not very well understood, and no treatments could be discovered. So fungal diseases continue to be a major threat, and you may wonder why we should discover structures of proteins to solve fungal diseases. And the simplest way to think about it is the structures of the proteins could be sort of imagined as locks, and then the drugs could be imagined as keys. Although not the perfect analogy, this lock and key model sort of helps you to appreciate why we need to determine structures. So we can say that if you know the structures of the locks or the proteins, you can then determine structures of the keys or the drugs that can better bind to the locks. And this is exactly what we do in structure-based drug discovery. We first determine structures of proteins that tells us the crevices within the protein. For example, in this case, if we make a drug shown in purple of a particular shape, it will fit very well in this conformation of the protein, but it will not work very well in this conformation of the protein because the structure is different. So by knowing the structural information, we can then go back and make drugs that will work better for a given protein, which can also help in not accidentally targeting other proteins which are unintended. So the goal was to determine the structure of ST2, and I first developed a new G protein, which we named as a mini G protein, which will allow us to determine the structure of ST2. And then I used a technique called electron cryomicroscopy, nicknamed as cryo-EM. And the way cryo-EM works is we first make a thin layer of protein molecules and quick freeze them at cryo temperatures, meaning very low temperatures, about minus 183 degrees Celsius. And this then is subjected to electrons, and from the scattering of the electrons, we get the two-dimensional projections of individual protein molecules. And using mathematical and computational algorithms, we can reconstruct a three-dimensional protein structure from multiple two-dimensional images. So we're taking pictures of individual protein molecules and we are reconstructing them into a three-dimensional structure. And this revolutionary technique has provided many insights into how proteins work at an atomic level. And a lot of this work was done at the MRC Laboratory of Molecular Biology, where one of my mentors, Richard Henderson, was awarded the Nobel Prize for his work back in 2017. Using cryo-EM, I went on to determine the first structure of ST2, and this is the structure of ST2 that we first determined in 2021. And it was very clear to us that this protein existed as a dimer, which was very striking. We were expecting to see just one GPCR, but what we saw was there were two GPCRs of the same kind. So it was a homodimer that was working together, and it simultaneously bound two G protein molecules, which was very surprising because this has been a phenomena that hasn't been previously observed. So what did we learn from the structure? We learned that this particular GPCR has a very extensive dimer interface. And most importantly, we learned that this GPCR binds to the fungal drug. In this case, it's called alpha factor, in a very different way compared to how human GPCRs bind to drugs. This is great news for drug discovery because the fungal GPCRs bind drugs differently, we may be able to develop drugs that specifically target fungal GPCRs, but not inadvertently target human GPCRs. And this is how structural information could be very valuable for such endeavors. I then was interested in determining the structure of ST2 in multiple different ways to understand how it gets activated, and I developed a new technique which we named passageway. I'm not going to go into the details, but what it allowed me to do is determine structures of STEA2 in multiple states. And in the end, we had 5 different structures of STEA2 in all the different states along its activation cycle, which told us that the activation mechanism of the fungal GPCR is very different from the activation mechanism of the human GPCRs. To put this into context, the first structure of a human GPCR bound to a G protein was determined in 2011 by the Kobilka lab, which led to a Nobel Prize in 2012. And since then, many structures of human monomeric GPCRs bound to G proteins have been determined. You don't have to be a structural biologist to appreciate that all these structures pretty much look similar to each other at a molecular architectural level. However, in the case of STEA2 dimer, we found that there were large-scale differences in that the entire G protein has shifted by nearly 58 degrees, and this allowed 2 G proteins to simultaneously interact with the GPCR. So this suggests that the fungal GPCR has a completely different mechanism of activation, as well as changes at the molecular architecture level compared to the human GPCRs. And this work was also featured on the cover page of Molecular Biology of the Cell textbook, which is used for teaching undergrads primarily. So where do we stand now? There are 10 different classes of fungal GPCRs, and the structures we have determined of STEA2 just belong to one class of fungal receptors. And there are still 9 other classes of fungal receptors with very important proteins, which we don't know anything about. And this is where the new tools we have discovered from this work, such as the mini-G proteins or the passageway methodology, could be used to determine structures of these unknown classes, which is ongoing work. And once we have the structures of these different classes, we can use all of them as targets for drug discovery. And already the structures of STEA2, which we have determined, have been used by multiple groups around the world to do computational drug docking to determine drugs compounds that would have the intended effects against fungal diseases. And we think there is a lot of potential for targeting fungal GPCRs, and we are just starting to unlock the potential of this work. So with that, I would like to thank my professor, Chris Tate, who was my PhD supervisor, in whose lab I performed this work. And thanks to CSAR for this opportunity. I'll be happy to take any questions.
So Vaithish, thank you very much. It's always interesting just to get a sort of narrative as to how these things happen, and you did a great job on that. It's way outside my special subject. I'm just looking to see if there are any questions that we can pick up. I mean, does AlphaFold have any relevance in terms of, is this a useful tool in the armory for sorting out new structures?
No, it is a very useful tool, but at the moment, AlphaFold doesn't have a lot of applications for membrane proteins yet. So AlphaFold was trained primarily with soluble proteins, and it does a very good job for the soluble portions of the proteins, but then for membrane proteins, it still hasn't reached a standard where it could be particularly reliably used. So there have been recent studies where they tried AlphaFold for many structures of GPCRs which didn't exist. And what they found was the models that they were getting from AlphaFold had critical differences in how drugs bind to GPCRs. And that's normally what you want to know from the structures, but it was being predicted wrongly. But it did do a very good job in many cases by predicting the overall fold correctly. So there are advantages with AlphaFold at the moment, but I'm very sure in the next 5 years or so, the models will get a lot better with better structures being available, and AlphaFold will have a lot more applications at that point.
Thank you. I didn't want to divert from your beautiful talk.
John, not at all.
Vaithish, thank you very much for a very interesting, very fluent talk as well. The structures that you're determining, I used to do electron microscopy, and they go so far beyond what I used to do. And I was just curious to know how long does it take you to go from, let's say, putting the sample, the thin sliver in the microscope and getting the representations to a determination of the structure? How much time?
Yeah, great question. So in my case, it took a lot longer because we had technical difficulties. So I won't go into the details, but there are some physical parameters that affected the orientation of the sample. And we had to develop new techniques to overcome those physical challenges. So in my case, for the first structure, we spent about 6 to 8 months. But if you had an ideal sample that you are applying on a grid, and if you want to go from that point to structure determination, this could be done in less than a month. So I know of many ideal cases for other GPCRs where a new structure has been determined in less than a month when everything goes very well.
So that's sort of dependent on the being able to get random orientations of the protein.
Yeah, so that's one of the main factors.
Yeah.
And also it's dependent on how stable the protein complex is and how homogeneous the preparation of the protein is. So sample preparation is the most important step. So some proteins are very well behaved and then other proteins are more fragile. So if the protein is very well behaved, then the technique now allows us to get very good structures in less than a month, which is a dramatic improvement in the field.
Great. Well, I think thank you very much, Vaithish, and